Hello, welcome to everyone interested in healthcare and medical science advances. I assume that's everyone. Today we are excited to share with you and hear about some new breakthroughs in healthcare and medical science that benefit patients, doctors, healthcare providers, and all stakeholders in medicine and health. This is the first of 5 videocasts. We'll be hearing from Dr. René Anand, who is the creator of the breakthroughs, and he is also the CEO of the new startup company, Neurxstem, in Ohio. Dr. Anand, I am very excited by precision medicine and its potential, and it's a very hot and exciting nowadays. It looks like you were the pioneer at the very beginning, creating and inventing the tools and technology necessary to make this a reality in the real world. And nearly 10 years ago, when you were covered by the mainstream media like CNN and BBC in 2015, you introduced this to the world. So first, can you tell us about the proprietary technology you have invented and that is protected with multiple patents?
Welcome, everybody. These are very exciting times. And one of the challenges back in 2015 and before that was we couldn't study brain disorders or diseases in humans. And it became necessary to create the tools and technology, which I will describe shortly, because the only things we could study was what happens to you when you had died, and we were able to get postmortem tissue. And that is very useful, but it was just one snapshot. You couldn't really tell what really started the disease, what are the factors that affected them, how did this get exacerbated into what we see now for disease like Alzheimer's, ALS, Parkinson's, autism. And so back in 2015, we were the first ones to disclose that we had made a complete human brain model, or they also call organoids. And that's very critical that we had created something that was complete. And that is because the human brain is networked across all the regions. And in the absence of that networking, you're not really going to get the full story of what is going on at the molecular level. So I'm going to share with you our exciting discovery that was covered back by the press in 2015. So Neurxstem is a predictive and precision functional genomic brain medicine company. And what I mean by functional is we don't sequence genes. We study them in their natural environment and how the genes are expressed. And by looking at comparing them between normal and disease states, we are able to achieve our goals of early diagnostics and treatment strategies for human brain diseases and disorders. Now, what I'm going to describe now is that we were able to engineer in 2015, which is when we made it public, a singular entity that contained all the parts of the brain. And yes, some of them are shown here, from the retina to the cortex to the spinal cord. And this has a US patent on it right now. And we find it fascinating that in 2023, the FDA Modernization Act actually arrived at the same conclusion that, in fact, the best models for human diseases were actually human tissues that were bioengineered, and we were ahead of them by almost 10 years. So I wanted to give you a little bit more specifics about what our model looks like. And because there was a lack of ability to study these diseases, other than in postmortem, as I mentioned, we engineered them from skin cells through a process in which we make pluripotent stem cells, and then using a proprietary process, make these brain organoids. We also call them NNOPs, which is Neurxstem Neural Organoid Platform. It's a platform. And fascinatingly, they contain all brain regions, all cell types, and is nearly 98% identical to the human brain. Nobody else has achieved this to this date. So a little bit more in terms of the details. Everybody likes to see data when you make claims like this. And here is the mini brain, or the organoid. And you can see the cortex, you can see the midbrain, hindbrain, cerebellum, blood-brain barrier, which most organoids don't have, and including the spinal cord. So we have the complete structure of the brain using immunohistological techniques. We also looked at the expression of genes for all the different regions, and you can see again, this is during development, expressed at different times in development, and you have all of the key genes that define the brain regions expressed. This is the spinal cord with all the Hox genes, and at this end is the cortex and the retina. We also have the microglia, which is the innate immune system. So it's a complete system with the blood-brain barrier, and it opens the door for doing absolutely astounding work that I will describe in many of the other podcasts that are coming up. And the question is, how do you know that this is reproducible and reliable for commercial preclinical IND studies that require FDA approval? And we know that by looking at the expression of genes across development. We're looking at 13,000 to 15,000 genes at maturity, and plotted on each axis are independent organoids. And you're looking at the expression of the genes here, where they have an R-squared coefficient of greater than 0.94 to 97 in most cases. Throughout development, D1, D2, D3 stands for days. These are weeks. At week 12, they typically mature. And we then went on to ask the question, could we do this for all of the different disease states, and which we have done, starting on the left-hand side, brain cancer. We've done a lot of work on aging, Alzheimer's, Parkinson's, and even progeria that we will talk about, as well as autism and schizophrenia. So the bigger questions that we need to get into will be shared on our next podcast with Kevin as our host. So thank you all for your time, and goodbye for now.
Thank you, Dr. Anand.